agriculture. And so what is the way in which we are going to do towards that? So last time we talked about the why we require moisture at the root zone to drastically bring down the water requirement of a plant. Next, I talked about measured moisture because we need to deliver it to optimize water use depending on the soil quality as well as the plant life cycle. So today we are going to come to the other three M's, which is microbes, mycorrhizae, and management. Next slide, please. So what is the goal? We want fast, healthy growth of high yield, high plant yield using minimal water and enhancing soil quality. This is because I think largely our drive for agriculture prosperity has been driven by petrochemicals. And petrochemicals require water, and therefore you went into a spiral where more and more water started to be used as less and less productivity of petrochemicals was happening, such as urea, NPK. And that has even made groundwater saline. So I think we need to get the entire cycle out. So the quality of irrigation should now serve soil health, not just plant growth and yield, which we have been either too stuck with. So we deliver more water at the moisture, as measured moisture at the root zone, and no other irrigation system does that. And that's our second point. The soil ecology is very important, which is the living organisms and the capillary spread of moisture, as well as microbes doing the job of soil moisture synergy, water soil synergy. Finally, if you want to get any theory into practice or even demonstration farms into practice, farmers should have the confidence, and therefore they should be able to recognize, measure the moisture status to plan their irrigation schedule, etc. Next. So I'm going to talk about how do you prepare microbe inoculant. We have done this after studying multiple practices in India, studied it scientifically as well, and therefore we found this as a very important way in which microbe inoculant can be locally prepared. The key part to understand is that while SWAR can be made available to you, I can send it to you, microbe inoculant has to be locally prepared because the microbes in the soil vary from different areas. It is different in Karnataka, even in Karnataka in different places, etc. So if they are not native, we will have a lot of invasive species, just as we have done with plants. But if we do that with soil, there is no way we can correct it. In case of plants, we can remove them, we can cut them, we can do things. But once foreign invasive microorganisms are introduced, we are going to face a disaster situation. So therefore, the microbe inoculant has to be locally made. The first step, as many of you should be practicing, is the preparation of Jivamrita, which is then applied within 72 hours. Next. I'm telling you how the microbe moisture is made. Then you have farmyard manure and red soil of equal quantity, right? So they are brought in and they are placed one over the other. So you have the farmyard manure as well as you have the red soil. Basically, it should be the soil of the opposite to what is there. Next. So you can see this from here on this. After putting Jivamrit, we put groundnut cake or any cake, any oilseed cake. And this has to be done under a shade. And it has to be done in a place where there's a lot of subsoil microorganisms. What we are doing is to make it inoculate using the organisms that are already within the ecosystem of that particular geography. And this is groundnut cake. This will provide the protein for the microorganisms. So you put one layer of red soil, then farmyard manure, Jivamrit, and then mix it up and then add groundnut cake. And so you keep doing layer after layer. And you can get to about 10 to 12 inches height, making multiple layers and all the time mixing and remixing them. Next. Then we also add one very important thing, which is the soil under a banyan tree. Now, why do we bring the soil under the banyan tree? You know that the banyan tree has the largest biodiversity of microorganisms. Our interest is to increase the number of microorganisms as well as its biodiversity. So therefore, the diversity comes by importing certain amount of soil under banyan tree. If you are making 5 tons of farmyard manure and red soil put together, add about 200 kgs of soil under a banyan tree. If not under a banyan tree, get it from a virgin forest area, because that is where the microorganisms have all its diversity. We don't have it in our existing soils. And then you put a layer, again you repeat this. I can send you a write-up, and it's easily doable, is what I'm trying to tell you. Next. So then at every stage, as I said, you put farmyard manure, then red soil, then Jeevamrutam, then groundnut cake or any oilseed cake, right? So these are the steps by which what you are trying to bring in is the microbes, both locally present, which you are trying to foster, and within the soil where you are making this has to be made under shade. And along with that, you are bringing in the microbial diversity, either through the virgin forest soil or through the soil under banyan tree. At every layer, you apply Jeevamrutam. You keep mixing it up, but keep piling it to a length of 12 inches max, 10 to 12. The length can be anything that you like. And this process will take you about 5-6 days to complete. So you are now ready, and then you keep on every day moistening it, preferably twice a day. Because the reason why we moved into measured moisture, not water quantity, is because moisture is the regime that microorganisms love. That is where they thrive. Whereas the drip irrigation logic of field capacity to wilting point is antimicrobial. Therefore, it is centered around the application of chemicals, and the biology of the soil is lost. So this is what we do here. Now, having got the microbes which are local and diverse, we go to the next step. Next. This is jaggery water we apply, which is basically Jeevamrit, as you know, right? Or jaggery water can also be applied. Basically, what you're trying to give is protein, sweeteners, and those things that entice microorganisms to start coming into this area and multiply. That's the whole idea. And this is based on multiple farmers' practices. We studied from Punjab to Tamil Nadu. We also compared it to Illingham's work in the US. And therefore, we have come to the conclusion that this works. Let me give you, we got this chemically analyzed. It's far superior to vermicompost. There's no comparison, although it costs almost the same. We got this live compost. We are now doing a partnership with the Central State University of the University of Iowa. They came to us finding that what we prepare is extraordinarily good. And they have taken the samples. We could test it only chemically in India. They could test it for biological. We find that this product is 100% organic and does not use any chemicals or petroleum products to enhance its effectivity. The raw materials that go into the making of the product is farm-based and locally available, sourced from plants and animals. The product does not use human waste or any recycled material from city waste. The availability of nutrients in the product are made available to the plants vis-a-vis a microbial process which converts fiber and other carbon compounds, which are then utilized along with mycorrhizae and rhizobium to enhance absorption of organically bound plant nutrients into the product. We found this product very effective in farm trials and field testing conducted in tropical conditions in India. We are now conducting a series of long-term experiments to find the mechanism of nutrient conversion and assimilation at the root zone of the crop. That's work in progress. It will take about 3 years for this to come out. In closing, the professor of biology there writes to me saying, at a time of global climate change crisis and other multiple anthropogenic activities has led to global food security threat and has enhanced the need for developing sustainability products that are de-linked from global threats and decrease dependence. This product needs more scientific development, and we are working towards it, and it is highly energy efficient, locally producible, and hence satisfies all the conditions required for a micro inoculant. Okay, so that's the testimony we have. It has also been validated by lots of farmers. I've seen trees which were not yielding suddenly to begin to yield, where the fruit size was low, the size went up. So I think living compost is what we call it as, is something that we advise farmers to make it. It's up to you to make it. We guide you at every step. Next. Now, the point is we now have microbes working, but I work largely in horticulture and forestry. So their mycorrhizae is very important. Mycorrhizae is largely fungal in nature. If you see a wheat crop, you don't need a mycorrhizae at all. You don't need fungal at all, and it comes up. Whereas in the case of horticulture, it has to be many times more. So there has to be more fungal in the fungi in the soil rather than even bacteria, etc. So I think for that reason, we have added what is the inoculum that can provide the mycorrhizae, right? So what we do is to take navdanyas, you can see it on the left, and from there we spread it on the soil, right? We cover it with some kind of hay so that this stays like that. Then we keep watering it, moisturing it. So these beds are mulched with dry grass and moisture. Inside are all the seeds of diverse varieties that we have put, which are all native, right? So we're talking about ragi, we're talking about jawar, we're talking about oil, groundnut, etc. That's what we do, and you can see that. So it will be a legume and non-legume, oil, etc. So that you try to bring the nature's diversity into the mycorrhizae that you're trying to foster. So that's how we cover it, and this is how it starts growing. It comes out of the dry grass. Next. This is how it finally looks. This takes about 3.5 weeks. After 3.5, so you put the navdanya around the day 10, and then from there it takes about 14, 15 days, and now it is ready. This you slice it and put it inside the soil, preferably along with swar. So when swar is embedded at the root zone, we make a larger place so that this can be put there. So what happens? You have moisture, you have measured moisture, you have microbes in the soil, you have mycorrhizae in the soil. This is the ecosystem for transforming Indian agriculture, and to make our agriculture rejuvenate rather than the direction we have been heading. It can produce a lot of food. There's no doubt about it. The soil is very healthy. If the soil is very healthy, obviously the produce will also be healthy. And that is what the consumers now want. I think that's the way in which swar delivers the task that we are talking about. Go to the next one. Do you have any questions at this stage, or because I'm getting into the management piece now?
No, you can first complete, then we'll take up the questions.
Okay. So I'll finish it in the next 4 or 5 minutes, because we must leave some time for questions as well. And people can ask questions even on my earlier presentation if later on found worth asking. Okay. Now, the point is that all studies show that drip is water efficient, right? Although it is centered around the around the use of petrochemicals. We leave that for a minute. Therefore, but in pharma practice, all studies showing that farmer irrigate much more than drip than what is recommended by the scientists. So, like for example, all of them open the drippers and let the water out. That is because between the what is done in an ICR lab or in a KVK farm vis-a-vis what happens in ground is the biggest challenge. Otherwise, there will be no water savings. Water savings will only be a claim. There will be no actual water savings because there is a big gap of 67% between what is theoretically recommended and what practically happens. 67% is equal to flood irrigation, which is what drip claims to be superior over. So actually we are back to square one, worsened by a regime that is in favor of the use of petrochemicals. Now, when in our system, I have promoted 4Ms. We also allow soil nutrients which can correct soil deficiencies. That is very important, like zinc, etc. Or what is the requirement of plant? For example, if you have some type of curry leaf plant, then it requires more iron. So you try to add that there is no problem. That also is delivered at the root zone. Therefore, you can use much less of them and get much better results and higher efficacy. Now, for example, since the key is the moisture level and that cannot be seen on the top, we use a moisture probe. There are two types of moisture probes. There's a portable one that you see in this picture where it can go up to 15 inches deep. We also have one which can go up to 18 inches and it indicates 4 colors. One would be red, which means absolutely dry, yellow, which means precarious, green, which means okay, don't worry, blue, which means excess. The idea is that the farmer is able to know moisture intensity and the moisture spread. Now, this will help the farmer if he does it over a period of time and in multiple seasons and weeks, they will get a complete schedule of what should be the watering that they should do. That will actually bring down the plant water requirement by 70 to 80%, which is the gap between what we apply as water and what some of the most developed countries like Israel have successfully demonstrated. Here, Israel has demonstrated higher yield, lesser water, but totally chemically driven or energy driven. India has to take a different route. We have to use less water more carefully, and we must improve the biology of the soil and therefore the yield. We are being a tropical country. This is something which is very required. So this is a moisture probe. There's another moisture probe that we also can provide. We don't make it, but which is embedded in the plant, next to the plant or a little away from the plant or the tree. There, the sensor has 3 levels. One level subsurface, second level root zone, third level drainage zone. So you put it actually 30 inches below, 40 inches below, and the lowest one. These 3 sensors send you the data, right? Up, of course, using IG, etc., etc. And you can get the data on your cell phone. We can provide a way in which you can switch off the motor from that. Now, this will give us a lot of data on how the moisture is behaving, both in terms of its gravity flow as well as its capillary spread. I think that is the way we are going to change the water regime into what we are talking about. So this is the way in which the farmer gets the confidence to practice what we have seen. And once he sees that for 2 years, then he is going to practice it. And therefore, we will have a real water saving, a real improvement in soil health. So let me conclude with Next slide, please. My internet connection is unstable. Next slide, please.
I changed, sir. I changed the slide.
Next slide, please. Next slide. Next slide. Okay, so what does SWAR deliver? I want to recap. Moisture at plant root zone to save water, measured to minimize water application, optimize planting. We cannot think of saving water without optimizing plant yield. The day the farmer sees minimizing water and optimizing yield, then he is all for it. She is all for it. Next, microbe inoculates for efficient plant use of the administered soil moisture and soil health. So that's what the microbe inoculum does. Then mycorrhizae for effective root, moisture and nutrient absorption with nutrient spend. Right. Finally, management is with moisture data-based decision making and farmer confidence. So this is the SWAR ecosystem that we work on. We provide guidance on how to make the living crop. You can see on the right side, SWAR is going down from the drip lateral. There's a big thing. The big places where we put the inoculums. 10 kgs per pit would be more than sufficient. And you can replace it after 2 to 3 years based on the type of soil that you have. You can also get the soil tested to be able to know what is the improvement in the carbon, etc. Soil deficiencies based on soil testing that you have done can be corrected also. We are only trying to move the petrochemicals out of this entire business. We cannot do it overnight, but let's do it over a period of time. So, next. Okay, I think that is done. My last point is this. I think agriculture rejuvenation is the major shift that is required. Are you able to hear me? Hello? Are you able to hear me?
Yes, sir.
Yes, we can hear you.
Right. So therefore, what is the major shift we must bring? Where I think our technology has a contribution to it. I'm not saying this is complete. I'm saying this is a strategic shift. What am I saying? I am saying we must not measure water. We must measure moisture intensity. We must not deliver water on the top or at one spot, but moisture should be diffused. It should be given at the plant root zone and diffused. Because we are in the moisture business, the world is in a water business. The moisture business is good for soil ecology, microorganisms. Right? Water concentration at one spot is excellent for petrochemicals. Next, this therefore you can bring a drastic saving in water, 60 to 70% easily. The next is soil health, because that is connected. That is where we bring in the microbes and the mycorrhizae, and that will improve soil level. Although it will take time before soils get into the biological sort of thing, but please remember, they spiral growth, whereas chemicals actually do the other way around. Right? They become autonomous to increase the diversity of microorganisms, fungi, etc. I think that's the direction ecological farming should move. So if the soil is good and the water is properly managed to suit the ecosystems that provide nutrient to the plant, obviously the plant yield will also be healthy. We don't have to be stuck with organic, permaculture, this, that, and the other. We know that this is a healthy soil and therefore the yield is healthy. So you produce healthy food. And that is what the entire country wants. And finally, we want to increase farmers' income. And because of all this, and being able to provide this to the customer of the quality, of the reliability of that they have, farmers' incomes will also increase. So I think that's the shift we have to make to regenerate agriculture. We are an NGO called the Center for Environmental Research. We have developed this over last decade, and I'm happy we've now got some traction, and I'll be happy to engage with each of you. And I'm thankful to Agri group to help sort of propagate this. This has been validated with over 400 farmers. We got the Pusa Krishi Award. ICR has validated it. University of Iowa has validated parts of it. I am going to collect the FICCI Award for water technology innovation around another 10 days. We got the 50 lakh award prize money from ACT Environment Trust. I think the point is that when you're trying to take a directional shift, it will take time, but we have to move along. And I'll be happy to help and work with you and your colleagues and your fraternity in taking this agenda, or even to clarify if you have any doubts and questions. Thank you.
Thank you, Mr. Gopal, for a detailed presentation. I'll now quickly take up some questions. We have not too many questions. So the first question is, what is microbe inoculant?
Okay, what is microbe inoculant? It is that you are interested in increasing the number of microorganisms in the soil. The number and the diversity. Microorganisms are multiple. We don't even know how many are there, right? Not the number, but just the sheer diversity of them. Because that's what happens in the soil. Please remember, unlike chemical sciences, biological sciences, especially related to soil, is very little known. Please remember, it is the microorganisms which thrive on our body also, right? But in the plant and in the soil, it's many times higher. What is an inoculum? Inoculum is like a dosage that is being given, which is so rich, right, that it will start accelerating its multiplication. All you're doing is to put a small piece because you can't fill it. For example, in Nigeria, we found that farmers put even farmyard manure at the root zone, right? At select places. They don't put it all over the farm. And they found some good results. So therefore, what we do is this inoculum is embedded on 3 or 4 sides of the plant or the tree. And from there, it starts multiplying and growing. Because that's why I said it has a geometrical progress. And the moisture regime is extremely suitable for microbes to thrive, especially because we're a tropical country. So that's the microbe inoculant. But the inoculant, as you said, I can make it in Hyderabad and send it to you. But it's absolutely bad because I will kill the organisms that are locally there. So therefore, you have to make it there. A lot of people said we will commercially make it. I have not agreed that it should be made commercial. People may make it. That's what vermicomposters do. Vermicompost is hybrid earthworms. Hybrid earthworms move horizontally. We want earthworms to move vertically. So therefore, there are multiple aspects of that. We don't have the time to go into it. So microbe inoculant, to cut a long story short, is an inoculum which provides diversity and intensity so that a natural process in the ecosystem that we are facilitating of moisture, measured moisture, they multiply. And therefore, the soil becomes rich in biology rather than anything else. Is that clear?
Yeah, okay. Okay, so the next question is, how does Center for Environment Concerns in Hyderabad exactly do? Sorry, what does your center do exactly do?
Well, what we have been exactly doing for the last 10 years is what? Before that, we worked on NREGA, Rural Employment Guarantee Act. We were into water conservation, participatory forestry, etc. But that's all history. Today, we are preoccupied totally with trying to transform Indian agriculture. In Bangalore, for example, we tried a small plot where I've taken a medium in the heart of Bangalore on the Yalahanka Dodballapur road, where I've shown how do you grow biodiverse plants there rather than what you have in the medians in Bangalore. And how do you conserve water? How do you use multi-story building, recycled water, treated water. How do you bring living soils there? And so that, Bangalore had a huge rainfall. The absorption in our place was very high. Whereas what you have there is absolutely invasive bougainvillea species. Even the tree species are, or Conacarpus, etc. So I think we are not understanding that we are stuck with breeding, but actually what we require is ecological gains. This was a small demo of ecological gains, and I'm hoping that it will gain traction, because what we need to influence is the ideas of people. Actions of people will follow later. So that is what CEC is currently preoccupied. We produce the SWAR system, that embedded moisture diffuser, and we make it available to farmers, to institutions, etc.
Okay. So I think you have answered the question just now. How does the center helps farmers?
Well, we don't help farmers. We are not project driven. Let's be very clear. Although we are an NGO, I've never went to a donor in my life. They came to me, right? Because we have something to offer. The farmer has to buy it, or the institution has to buy it. A lot of my customers have been NGOs, right? I give them free is the knowledge on how to make the microbe, but the moisture diffuser, I make them. It costs me money. I'm not giving it free. It has to be purchased. So is also the moisture probe. There's a moisture probe. I make it available for 1,400. If you want totally embedded ones, sensors, electronics, Wi-Fi, it may cost anywhere from 12 to 15,000. I mean, the monthly fee payable to the telecom companies. The choice is yours, but we are not a implementing agency saying, here, give me a project, I'll do it. We will do it. Like for this project I did in Bangalore, if somebody said, I have a dream, do you have one? I said, yes, I too have one. Let's do your dream. So that's why I did that median. Why did I do median in Bangalore? Do you know Bangalore has a total coverage of 75,000 hectares, the Bangalore city, and its neighborhood. Do you know 1,450 hectares come under medians? Imagine the potential of that 1,450 hectares to harvest ecological gains, bees, butterflies, carbon sequestration, enormous. But that's not the direction we are heading to. So therefore, that's what we do. Where our interest is to learn, our interest is to enhance. So we do that. But otherwise, if you are an institution, please let us know, talk to your farmers, explain to them, and take it on further. We want to transform Indian agriculture. And we are not a profit agency. We are not. But that doesn't mean we will pay the cost. That is not possible.
Okay. And now we have the last question. Please let us know a little about your career background.
I have a master's in business administration. Then I studied at the Indian Institute of Foreign Trade. I studied international trade there in Delhi. My first job was with Shiv Nadar, who started HCL. In fact, HCL was not even started then. It was just registered. So I worked with him for a year. It was a company which he founded called Microcom, and I was working with him. But I was not interested in furthering a corporate career. So I came back to Hyderabad and I joined the Administrative Staff College to teach management there. I was doing that in 1980. Then I thought that teaching corporate managers how to manage will not change the world. It could improve their business. But to change the world, we have to bring management concepts into rural development. So therefore, I decided in 1980 that I will work for rural development. Initially, at that time, the laws favored corporates to donate. So I started it with a company. My water was my first thing. I believe that time that water is central to successful farming. Please remember, it was in the 80s. 3 technologies came in: borewell, high energy, drip. All of them came around that time. So I got excited saying, yeah, here is a solution to the farmer. But within 5 years, I found that the farmers were actually losing because they were poor farmers. They were small farmers. groundwater table was depleting. Their yield was low. And therefore, I found that it was a zero-sum game. So I went off to work on dryland agriculture. Not much I got. Went off to the United States. I spent some time there. Then I was teaching in Europe. Then I came back to India. I published some books on foreign funding and the ideological issues thereof, dependency thereof. And then I worked with a consortium on integrated pest management, trying to mix, both chemicals and organic methods. That we did it for 7, 8 years together as a collective. Then I got very actively involved in the National Rural Employment Guarantee Act. I got involved in the act because in 1984, I was one of the 100 students from youth from Hyderabad who went to Delhi demanding the right to employment, the cycle. So when it came, I got very excited. We did a lot of contribution. I've written a lot of books. Manmohan Singh released 2 of those books. I was made a member of the Central Employment Guarantee Council. I chaired the Committee on Works. And then I realized that water conservation was #1 in our region. At that time, the Tata Institute of Social Sciences, Bombay, invited me to be a scholar in residence. They told me, you come there, be with us for 2.5 years, and try to bring your practice into theory. Right? That was the mandate they gave me. They gave me a house in the campus. I liked it because Raj Kapoor used to live next door to that, and I'm a fan of this. So I was there between 2010 and 12. I tried to bring as much of practices. It's not so easy. Pedagogy has to change. Case studies have to change. Approaches have to change. Otherwise, they were just downloading it from Google. What is this? What is that? Etc. And at that time, I also had some spare time. I realized that this way we are not going to solve the water problem. Water can never be self-sufficient. You have to work on what can be done to reduce water usage. So I went to water efficiency. Then I worked with farmers. Then we came up with saying, no, provide moisture at the roots. So I did it with pots, etc. There were a lot of problems with it. Then I shifted from one to another. Then I found, yes, I can save 50% water. Then I realized it's not enough. How do I optimize water? I'm not in the water saving business. I'm in the business of farmers getting better income. So optimize water. That became the measured irrigation. That's where I use KC values. Nobody is using them. They are actually most of them. I have programmed the entire KC values. I can tell you what should be the KC value. Then I realized this is not enough. We should think of soil health, which is basically microbes. So I worked on microbes. I was a consultant to Ramoji Film City for promoting organic. I told him I don't know organic, but he said, no, you do what you like. At that time, we developed this living compost. And then I realized that I'm in horticulture. So I added mycorrhizae. And then by that time, digital technology, sensor technology had advanced. I have confidence in them, though all of them are still yet imperfect, I must say. So I put all of them, and that's where I am now. Okay. Thank you.
That's a very good journey, sir. So, okay. So I don't see any more questions from participants. So now we have come to end of question round. On behalf of agricultureinformation.com, we like to thank you for a very detailed presentation and answering all the questions. And we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Thank you.
You're most welcome, sir. Thank you.